Okay, so we're going to take up today here the different bacterial cell structure as our laboratory activity. So bacterial cell structures, or simply you call it one as the bacterial appendages, has been utilized by some bacteria as their mode of their locomotion for their protection and many other purposes. So the first one, we have here the flagella. So flagella here, okay, this is a protein structure that try to surround here the bacterial cell. Okay, and this would have here antigenic properties. They could be identified here by our serological typing. So basically, the flagella serve here as your H antigen that could be detected here by our anti-H anti-sera. Okay, so flagella is being utilized by many organisms here which are motile as their organ for their locomotion. So better way of visualization of your flagella, you could use here the grace na stain. So that one is a very specific here for the staining of your flagella. Okay, procedure for this one, just need to examine under the microscope and try to draw here the structure of your Klebsiella pneumoniae, your Salmonella typhosa, and we have your Proteus vulgaris. These are the organisms which contains here the flagella. So another one, aside from drawing here the two, you should also draw here your flagella according to their location and to the number of the flagella. Okay, there's a microscopic field that you need to draw here at trichos. So sa atrichos, I mean to say, so bacterial cell lang, wala siyang flagella. Monotrichos, those are the organism here that contains only one flagella in one end. Lophotrichos on the other hand, so nasa isang pole lang o isang end lang pero madami na flagella. Amphitrichos on the other hand, so dalawang end, one here, another one on that. Okay, would have here the flagella on both ends. Peritrichos, the entire body or the cell of the bacteria would being covered here by our flagella. Okay, the next bacterial appendages, we have here your capsule. So the capsule here, so this try to surround basically your bacterial cell wall. So it could be in the form of so that's your glycocalyx. So that's maybe in the form of your capsule. This is a polysaccharide capsule. The capsule here serve many purposes. So number one, it try to have here antigenic properties. So it would have identified here as the K antigen, capsular antigen, or the VI antigen. Still, that's your capsular antigen pa din. Okay, so capsule here could be differentiated from our slime layer because the capsule would be firmly attached to your bacterial cell wall. That one is organized. Compare with your slime layer, that one is not organized, and at the same time that one is loosely attached to your bacterial cell. Okay, so the capsule also here, the polysaccharide, specifically the polysaccharide capsule serve here as the basis for the Lancefield grouping among the Streptococcus. So ang strep natin, we could classify that one as to their Lancefield grouping. At ang Lancefield grouping nila, kung bakit sila classify based on that Lancefield grouping is based on their polysaccharide capsule. That's the basis for that. Okay, another one na purpose ng capsule natin. So it provides here the virulence factor for your organism or bacteria. So because of the capsule, the organism here without the capsule try able to prevent the phagocytosis because they are being covered by the capsule and therefore the phagocyte could not recognize your microorganisms having the capsule. Therefore, try to resist here the phagocytosis. Okay, procedure for this one, just need to examine here the different organisms which has the capsule. We have here your Klebsiella pneumoniae, your Strep pneumoniae, and we have your Neisseria meningitis. So, pag nag-draw kayo nito dapat, like, if the organism here is a gram-positive, so dapat blue ang color nito. Pag ang organism natin is gram-negative, then dapat red ang color nito. So, capsule would be identified here as parang clear area. You need to draw here parang may space in between your bacterial cell. Tapos, okay, yung nag-cover yan, sa kanya may space. So, become here the capsule. Okay, let's try to surround here your bacterial cells. So, that's your capsule. Okay, so another bacteria appendage here would be your spores. So spores here would basically, this is a refractile oval na structure found here in the cytoplasm bacterial cell. So your spores could be found here at the middle portion, the central or subterminal, medyo malapit sa terminal end. And we have here the terminal. So terminal spores most likely found here on the terminal end. Just like your Clostridium tetani, nasa terminal end siya. So there are two forms of the spores here. We have your endospores, and we consider this one as the inactive, the dormant. And this one is your asexual na spores. So pag sinabing asexual, so this is not used here for bacterial multiplication. On the other hand, once your bacteria having the spores has been exposed to your harsh environment, including here high temperature, desiccation, drying up, change in the pH, or even exposure to your radiation would convert that one to become here your vegetative form. So the spores here used by the organisms as their protection. Okay, so there are two species of or two genus of the bacteria having the spores. One belonging to your gram-positive na Bacillus. This includes your Bacillus anthracis, Bacillus cereus, Bacillus thuringiensis. You could also have your Clostridium na genus. Ang Clostridium na genus natin, they are gram-negative. So they could be formed here in the Clostridium tetani, Clostridium difficile, Clostridium botulinum, okay, and other form of your other species of your Clostridium. Okay, procedure for this one, just need to draw here your Bacillus anthracis and your Clostridium tetani. Okay, and then try to draw that one according to their gram staining reactions. Okay, pag gram positive, so blue ito. Pag gram negative, red. Okay, and then try also to draw here organism having the spores found in the central area, subterminal, or even the terminal area. Okay, the next one, we have here the granules. So the granules serve you as a storage component of your bacteria. The granules here could be in the form of your polysaccharide granules, in the form of your storage form na glycogen, or the granules could also be in the form of your lipids, your poly-beta hydroxybutyrate polyphosphate. So example the granules, we have here the metachromatic granules or the Babe's Ernst granules among your Corynebacterium diphtheriae. So ang kanyang granules is actually a form of your polyphosphate. And much granules found here on your Mycobacterium tuberculosis. This is gram-positive ang ating Corynebacterium diphtheriae. This is your acid-fast na bacilli, so this will be color red. So just need to draw here for this activity. Just need to draw here your Corynebacterium diphtheriae showing the metachromatic granules. And it should be draw here as the color blue. And Mycobacterium tuberculosis as color red because this one is the acid-fast bacilli. Okay, so thank you.